Hello everybody and welcome again to PASCO Laboratories. I'm JP, and before I tell you what the P stands for, let me tell you in advance, you're going to want to share what you see today with every middle school, high school biology, agriculture teacher, because we're going to do some very interesting demos that you can get your kids talking about right away when they might be sitting at home watching and thinking, or you want to get them engaged in thinking. We're going to help you. But before we get there, I just want to hop onto some quick viewer mail that kind of helps us get into our discussion today. And I want to start with an email that we got from Jack. You know Jack.
Absolutely.
Right. You know Jack. Jack in North Carolina writes, "My car is PASCO powered." You can see his car here on the slide. "My students laugh about my license plate, but when we start doing experiments in physics, they understand why I love PASCO. Can you send me a bumper sticker too?" Well, Jack, absolutely. We're going to be sending you a bumper sticker just like the one you see here. And thank you so much for sending that picture in. And to anybody out there, if you've got great things that you're doing with PASCO and you send me the picture, we use it here, we'll be happy to send you a bumper sticker also so that you can adorn your car just like Jack did. And I think you have some advice for Jack when it comes to that car.
Say, Jack, I know we're going to send you a physics bumper sticker, but please don't use your car as a collision cart in your own experiments in that way.
Right, we've got something for that. Yes, exactly. Now, we have another interesting question. This came in from Ernesto and his family, and it comes from Ernesto, and he says, "I was with my family in a park over the weekend, and afterwards I asked my kids to wash their hands." And we're doing that a lot now, Roger, right? So Francisco, his son, asked how hand soap might impact the environment. Do you have a soap sensor? Well, no, we don't. We don't have a soap sensor, so what we're going to send you instead, Francisco, is some of our great little STEM guy. Thank you for being a STEM guy with us, and if you have questions like those, parents, send them on in and let's think about them. We'll get some of these STEM guy stickers sent out, but we don't have a soap sensor, but it did get me to this point where what the P stands for, because the P stands for ponder, which is exactly what we're going to do today, Roger. We're going to ponder some things. And so as a quick introduction, for those of you who may not know me, I'm Dr. J.P. Keener. I'm the former director of STEM for the State Department of Education. And today I'm here with Roger, and we are pondering. And so, Roger, as we get into pondering a little bit and we think about Francisco's question, and he's asking questions about soap and the environment and how it might impact the environment, we sat and talked, and you said, I've got some neat things that people can show to our folks at home and that they can actually do and demonstrate, and you said let's do something with milk and get a discussion going.
Absolutely. As long as you have some things at home that are pretty common, I think you'll find that something like, just a little food coloring, and I've got some milk here that I've already poured into a bowl for us to get started. I'm going to just put a drop or two of the food coloring in different spots around the inside of this bowl of milk. You can do it on a flat plate. Just something that holds maybe an inch at the most is all you need in a nice flat surface. And I'll get different colors because that's what makes it kind of look cool. And the question really was centered around soap, and soap is an interesting molecule that has got a long arm to it that's sort of more like oil. It doesn't mix well, but then on the front end is like your hand, and so that end is got a plus charge to it. It's ionic. As a teacher, you'd know parts of those, but maybe as a student you don't think about that. And that hand can grab things that are like the colored molecules in here. The long part of the arm, though, does like the fat inside of the inside of the milk itself. So
we'd be talking about like the hydrophilic part and the hydrophobic part, right, if we were into our discussions.
Right, and our hand loves the water, hydrophilic loving, and then our arm likes the oily parts. And so I'm going to take some soap that's on the end of this Q-tip, and I'm going to put that right in the middle, so if we get a good overhead shot there. And let's see what happens as I put this into the water source.
That's very active, and there is nothing mechanical happening here. There's just that soap and that milk, and there's some stuff going on.
As a matter of fact, if you're doing this at home, that we have provided the little video, if you want to go ahead and roll that, is that, sure, okay, we've started to understand that soap can pull these colors aside. We're going to look now in 3 different kinds of milk, and that first one that I'm demonstrating here in this little video is the skim milk. Skim milk we'll start with, and I'm going to put a couple drops of Dawn onto the Q-tip, and we'll see a milk that has only 5 grams per cup of fat inside of it. It's got a little bit of fat in it, and when you put that soap in, it's going to have less fat.
And what I'd be doing with my kids at this point, if I were pushing this at home, I'd be asking them, what differences might they think they're going to see in these 3 different experiments. So this is skim milk,
Yep.
and we're watching what happens here. And you're going to demonstrate whole and a half, and I'd be asking my students at home, what do you think is going to happen?
Exactly.
And get them engaged in that discussion.
So when we move to whole milk, which has 9 grams, if you look in the back of the milk carton per cup, you can see that looks a little different. And that was what we started with, was the whole milk. And then we think, oh, there must be something even with more than that, and half is a good example, or cream would be great. So you can try these again at home, and you can try different kinds of soap as well. But we're sticking with just one variable at a time, that we have this great cream source.
And so this is our half and half.
Half
right?
and half is 24 grams per cup. And we'll let you be the judge to explain why you think these interact in a different way.
And so we can see this. This is in the case of the whole milk. It looks like this is just. It looks like it's just going to go on forever and ever and ever.
Right.
This just continues to move and go, and there's a lot of action there. So to me, just looking at it, between the skim and the half, I'm seeing a big difference. But
Absolutely.
this is a great qualitative opportunity to take a look at some data. This
Yep.
is qualitative data,
Yep.
and explain it. Folks at home, you're welcome to use this video if you want to engage your students at home in this type of discussion, even when it comes to like the scientific method or the processes of science. Feel free to use this video, or you can just do this demonstration on your own. You see how easy it was, and Roger did it here. But what we got into with that was now kind of thinking about back to Francisco and his question. Now I see what that soap did in this biological mixture, milk. I saw what it did. But I go back to Francisco's question and these 2 environments, and I ponder, our P word, I ponder the impact. In environment A, we're looking at a pond, and in environment B, I'm looking at a fish tank, and I ponder if soap would have an impact in those environments. And we could actually explore that and take that a little further. Before we do that though, Roger, I did want to say that that was actually Francisco who sent in that nice pond picture, and we see him here sitting off, sending us that slide and pondering what soap might do to water. So let's find out. Let's do some soap and water things that can help answer Francisco's question. What do you want to do?
Absolutely. And so you've seen that molecules moving across areas is the important thing in biology. Nutrients have to get to some place and move into an animal so that they can meet their needs. And we're going to take a look at a source of pond water. So I've gone off to the creek that's just outside here, and I've got in here 3 different kind of measurement tools that we use. Now, the first one that you're going to see is this side here, and that's the oxygen that's in water. And of course, just like fish and people and animals in the air, fish have to breathe the dissolved oxygen there. That's what their gills do. And your question was valid. It was a great question. And we're going to just put some, put some soap into that same water.
So before we put that soap in there, folks, ponder what's going to happen when Roger adds soap to that water. What happens to the dissolved oxygen?
And that's the first thing a fish needs, is that oxygen. So that's the first question we're going to see. Will it impact the ability for that animal, whether it's a fish or an insect or whatever else lives in that water? So I'll pour a little bit in, and
so we started at 19.34%, and we see that, well, Roger, I'm not seeing any change at all.
as a matter of fact, we could sit here a while, and I don't think it's going to change much at all. And so in that respect, what happened for the use of soap in the environment?
So soap has absolutely no impact on the oxygen in the environment.
So fish, insects, and those things can continue to breathe the way they had before.
So are we able to answer Francisco's question at this point then, like, hey, soap is fine in the environment, don't worry about it?
But there are other kinds of properties
there's more
that
to the
soap
story, so
can impact as well.
don't answer yet. Let's continue to ponder a little bit further because we can do some more analysis. What else do you want to check?
So the next probe we're going to look at is the conductivity probe. And usually all water sources have a little bit dissolved in them. So you might see something like a 300, 200, 400 for nice, clear, fresh water. That means a little bit of salt has dissolved in it. And when salt is in there, salt breaks up and can move and take charge with it. So we've got a
So we're
conductivity
here at the pond
sensor.
water, and I brought up that data here on my computer. I'm using SparkView, the same software that our folks can download for free. We're using your conductivity sensor, and I'm measuring 508.3. That's microsiemens per centimeter, and that's its current conductivity.
And that's good water. You typically non-salty water is going to be, like I said, 300, 400, 500 is a value that we'd expect from water that you'd find as an open source, like in a pond or a creek, that's in really clean conditions.
So as you add the soap, I ponder, what's going to happen to that conductivity?
So we
And
poured.
I'm watching my numbers, and well, heck, before you even
Stir
stir
it,
it, before look,
right.
those numbers are, wow, wow,
And now,
wow. Okay, so dramatic increase here.
absolutely. And if you've ever washed dishes or had your skin into water before, you may notice that, especially if you live near the ocean, that your water, your skin will be impacted by soaking in those, and that's that osmosis process driving
Diffusion process.
diffusion process driving water into your cells. And so, if you lived in water all the time, that diffusion process is pretty darn important to govern a part of a major organ in your body. And so we have to be a little careful, because soap, even though it's got that nice long grease-loving part to it, it also has a salty part to it. It likes the water and salts, so it does increase the saltiness of the water, which we saw.
I see that, and we see that here. So from what I'm thinking about now, we went from 500 to over 1,000. We doubled what I would almost imagine, like if I'm trying to breathe, I'm a fish in water, it would be like trying to breathe smoke. This is almost like particulate matter.
Yes.
These are dissolved things in the water, which make it difficult to live in that water.
Right.
True?
Right. Any surface that's conducting it is going to change the way the water moves in and out of those cells, and it might either become swollen or it might become pruny. And it will cause that to be not
pruny.
It's a real official term.
Okay.
Exactly.
So what we've seen so far then, Francisco, is that while it didn't impact the oxygen, it did certainly impact the conductivity, which is the amount of dissolved substances in that water. So this looks to me like it's not a good thing, but Roger wants to take it a step further.
Now, a conductivity sensor is great because it can just detect that things are dissolving and breaking apart in water. But if we go to something like pH, we can determine what is in that substance that breaks apart. A pH sensor is great at measuring compounds that have H in them or that like to steal H from water, acids or bases. And so acids and bases are critical because almost everything in your body is driven by stuff that's normal, sort of pH 7. So when you look at this, water that comes from outside around here has got a little limestone in this region.
So I've got it hooked up. I hooked up our SparkView. I've connected now to your wireless pH sensor.
Yep.
And I'm measuring our pH from your pond water at about 8.3.
And normal water is going to have no acid or base in it. It's going to have around 7. 8's okay because in nature there's rocks that can dissolve and create that sort of 8. But in that range, fish are completely comfortable. Animals live in that kind of water. So let's add our soap just to see what that does for the acidity portion.
Okay, and I'm watching my number, and
if I just give that a little bit.
8.4, okay. Oh, wow. All right, we're seeing we went up quite a bit. Actually, we started at 8, we're up over 9. My pH has gone up, which means it has become more basic.
That's right.
And I would imagine that's not good.
Absolutely. Base on your skin, if it's strong, like in drain cleaners or those things, cause wounds. Your skin's integrity starts to fall apart, proteins, hair, that kind of stuff. And that's why you put it into a drain to get that grease or hair out of the way. So that is not a good thing, even though fish don't have hair or those things. The skin itself will also react to that same level of base, and it will cause damage, especially if they're young, like eggs in that sort of cycle, that you'll cause damage for those. And so we've seen 2 different pieces of evidence that say soap is not.
Could be very dangerous to the environment. And of course, we know a little bit of soap in a fish tank will kill a fish, but now we're getting a better understanding of what's taking place. We looked at the oxygen, we looked at the pH, we looked at the dissolved materials that are in that, and we were just looking at the water. This is like in a water environment, but we know there's more to an environment. When I go out walking, I'm not in the water, I'm actually walking on what I call dirt. And I think a lot about the dirt, and of course we say dirt's not living; it would be abiotic. But
yeah,
that's
we
not
wouldn't
totally
have to worry
true,
about
right?
pouring soap into it, for instance.
Who would care? It's just dirt.
Yeah.
But there's more to it than that. Right?
Absolutely. And the one way we can determine that is even in conditions like in California, it's really dry. You may think, oh, there's not much going on in that dirt. So what we've done is we've taken 2 samples of dirt that we've just collected from right outside the building, and we've added a little bit of water to one because it's been a long dry season here, which is typical in California. And we've added some water just to make sure anything that's living in there and eating and being active, like worms or insects or even just some small microscopic sized materials, are completely living, eating, and when they eat those things, just like you and I, they're going to breathe out carbon dioxide from the food that they eat. And so we're going to put 2 carbon dioxide sensors in there, and we're going to compare that, which is dry, and
okay, so
a
you just
little
added
bit
those.
of water.
Let's take a look at the data then. We have just connected, and let's see. if I can't see what's happening with that data? So to me, we've just started it, and it's looking to me like, well, Roger, to me it looks like they're identical. I don't see any difference. But oh, wait a minute.
Yeah. And carbon dioxide's a little heavier than air, so it takes a little time to fill the bottom of that container. Now it's just getting up to where the sensor is.
I see what's happening here. So looking at this data very rapidly, comparing these two soil samples, I'm able to see that the dirt is actually doing something. I'm not able really now to say that dirt's dead.
Right. It's got some things that are living in there and breathing. And just like things that are in the water, soil has microbes and things that if you're going to put things like soil or like soap or other kinds of contaminants on the soil, you may cause damage to them, and this dirt won't continue to function in its proper, it's got a purpose, and that's to purify stuff and to take nutrients that are dead from old plants and make them the raw materials that plants can use to grow.
To grow new things. And so we've actually answered Francisco's question now in different components. We looked at water, we looked at soil. We now take a look at the question I throw out to our audience to ponder. We've got two different environments here, environment A, environment B. Environment A is just a forest where we've got dirt that is not dead, and we've got environment B with a potted plant with dirt which is obviously not dead. And much like I would do with my students at home in a distance learning situation, I would ask them to ponder the consequences of what soap might do to that environment. And without giving the whole answer away, Roger, what do you ponder might take place?
Well, I do ponder that the soap will cause some changes in the small biome. So if that's gone, your job is to take living and dead materials to recycle them for the plant, and eventually the plant may suffer. That'd be my guess.
So that's a great thing to ponder. And so we're actually showing you great demonstrations you can do at home using some very simple sensors. In fact, in this case, Roger was able to use dissolved oxygen, conductivity, and the pH sensor. And he used those, and what a great demonstration you've done there, Roger. But we've got something exciting that you've actually done for our folks at home.
Absolutely. If you're a teacher, or if you're in home and you're learning in this kind of an environment, where can I get a small set of equipment that really helps you investigate a lot of these living systems? We've put a small kit together that includes these types of probes so that you have a number of experiments. And so these we call our starter biology student lab kit, and that you can get a lot of the probes that we had used today, from conductivity sensor, the pH sensor, and we have a list of materials that you can go through as part of that study.
And when we talk about this, you know that I like to call it our 10, 10, 10, 100 package. The 10, 10, 10, 100 package, because it's got 10 ready-to-use labs that many of which you've already written, Roger. We've got 10 ready-to-use labs, 10 student data files, the type of data file that you saw collected here when we were doing pH or conductivity. That data can be given to your students so that they can look at it. Back home with live, not live, but real analysis capability, because they have the software that they can download right away. And 10 data collection videos, just like you're seeing here. Roger does many of these videos where he's actually collected the data, shows that data, shares that data, and of course 100% of all of the teacher resources that are available. And when we talk about the types of labs that Roger and our team here at PASCO Laboratories have written, we have a lot of different types. We have everything from enzyme action to osmosis to acid rain, cellular respiration in yeast, and even some computational thinking that gets into using Blockly as an extension. And that's a really great start with the labs that come along with the 10 data sets, the 10 data collection videos, and 100% of the resource. And that's why I call it the 10, 10, 10, 100 package. But we don't just do it for biology, do we?
Right. Here you've got a biology set of probes, but there's also agriculture sets of equipment that you can investigate these kinds of things that make living possible.
Sure, and this is actually, this was the ag sample that we were actually showing. Biology sample here. This is the ag kit that has every sensor that you use today for agricultural study. And this is also a 10, 10, 10, 100 package. Why is it the 10, 10, 10, 100? Because it has, Roger, 10 labs.
10 suggested labs.
And look at the labs that we have here. Everything from soil quality to plant pigments to greenhouse gases, diffusion, and a Blockly extension if we're doing some computational thinking, plus 10 data sets and 10 collection videos. And who's pictured in that video, Roger?
Looks like myself.
It looks like yourself because you've done many of these great videos that we've put out there to our folks, that we share with our folks, that we use with our folks all of the time, and of course 100% of our teacher resources that are available. So if you'd like to do these great demonstrations, you can start off with milk and have a great discussion, or pick up one of our kits that we've prepared for you. We have them available for you. If you remember today's word, what's our word today?
Ponder.
And if you call Isaac, who's Isaac? Isaac's our director of sales. He is our cat in the hat when it comes to making deals, and he wants to make a deal for you when it comes to these great kits that you could use even at home to do simple demonstrations for your students at home. Great kits, great example of how we can assemble something and do some great science like we did here. Call Isaac. Tell him you ponder the great price that he's going to offer you, and Isaac's going to give you a great price if you use the word ponder, and ponder the good prices there. And if you're not right here in America, don't worry. We also have wonderful opportunities for all of our international friends out there. Just take a look at our website, find your international partner, ask them, ponder with them how you might get these kits, and they'll be able to get them to you. And just as a shameless plug, Roger, I just want to throw this out there, that we also have the PASCO Academy. And so if you don't want to do any of this teaching and you want to see Roger every day for 15 weeks, you can, because he's going to be available for 15 weeks starting September 1st in our own PASCO Academy, where he's going to be doing demonstrations just like this, and even more exciting, every week for 15 weeks that you can send to your students. Let Roger do the instruction, let him show you the lab, let him share the data, let him do the analysis. You can just kick back with a cup of coffee and let Roger do the work for 15 weeks, and that's our PASCO Academy right there on pasco.com/PASCO Academy. And of course, all of our live events are recorded for you. So feel free to check out anything that we talk about every week. We really hope you send in your pictures. We hope you send in your questions. We hope that we can continue to send out our bumper stickers and engage with you, and we really appreciate you guys joining us.
And we're excited to have you, if we can help out in this time where it's crazy kind of teaching situations, to have an example set of data, an example of us showing the lab when you can't get your kids in the lab. So getting either a demo kit so you can show those on a Zoom meeting when you meet with your kids or whatever platform you use, and having, if you like the PASCO Academy, just to have the data and the labs all provided so that you can put those in your learning management system, would be a great way, I think, that we can help out while we're getting through this all together.
And we are, Roger. We're getting it all together. We ponder how we can help you, and we're here to do that. And so until we talk to you again, we wish you all the best of luck in your classroom, great teaching, and of course, good day.